Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled, How to Better Optimize an Oncology Drug Discovery Program. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout this presentation by using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, and that's found on the right-hand side of your screen. If you require assistance, please contact me at any time by sending me a message using that chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available to you for future streaming on xtalks.com. At this point, I'd like to thank Oncodesign, who developed the content for this presentation. Oncodesign is a biopharmaceutical company dedicated to precision medicine, founded in 1995 by its current CEO and majority shareholder. Now it's my pleasure to introduce you to your speakers for today's webinar. And first, I'd like to introduce you to Olivier Duchamp. He is the head of the In Vivo Sciences Department at Oncodesign BU Service. He was at the founding of the company and for the last 26 years contributed largely to its development and innovation in preclinical models and pharmacoimaging. Olivier is also the coordinator of Innovative Models Initiative, a French consortium dedicated to the development, the characterization, and the commercialization of new preclinical models in oncology, including PDX models and humanized mouse models. He is the co-author of several publications about PDX models. Now it's my pleasure to introduce you to your second speaker, and he is Jean-François Mirjolet. He is the head of the In Vitro Sciences Department at Oncodesign BU Service. He has been working for Oncodesign for 19 years, going through various positions from study director, technology director, to his current role managing both services and the R&D related in vitro activities. He obtained his PhD from the University of Nancy in cellular pharmacology of anti-cancer drugs, followed by postdoctoral position at the IRIBHM, Brussels, Belgium, in chemokines and chemokin receptors implication in cancer cell migration. Now, there's Jean-François and there's Olivier. Thank you very much, gentlemen. Now it's my pleasure to pass over the mic to our first speaker, and that's Jean-François. So Jean-François, when you're ready, you may begin.
So, hello everyone, it's my pleasure to be here today. So, first I would like to introduce a bit about Oncodesign service. So, Oncodesign service is providing support in various disease fields such as oncology, immuno-oncology, inflammation, inflammatory disease, and infectious disease. Our services are based on various scientific and technological platforms. Among them, we have medicinal chemistry platform. We have recently developed bioinformatics support and artificial intelligence capabilities to support analyzing clients' patient database, and for sure our core facility is pharmacological analysis of compound. The offer we want to talk about today is mostly related to our impact offer. And impact is really our offer to support pharmacological development from start to the end of compound development. And we would like to focus today on oncology impact offer. So I'm here to present our in vitro capability, and before doing so, I want to introduce a bit the recent move in vitro pharmacological analysis. First, in the early age, we used and mostly used two-dimensional cell culture system, and this system has been evolved through mixed culture and compartmental culture such as Boyden chamber. However, we all know that this still used two-dimensional culture is far from disease modeling, so it has been evolved to three-dimensional cell culture, so spheroids, or even more better, more improved systems such as organoids, which is directly derived from patient material. In addition to these two systems, two-dimensional and three-dimensional, there are now a lot of devices that can help to model disease and to understand cells interaction, which is at some point the key of the treatment benefits. And there are now a lot of microfluidic devices that are here to support disease modeling, and now we can also benefit from a bioprinting system. The key parameter in deciding to move to 3D are the ability of 3D system to really model what we have in vivo and for sure in human. 3D gives us the ability to work with gradients or a lot of things, meaning oxygen, glucose, proliferation, and lactate production, DNA break, which are all the parameters that really represent the disease in their clinical situation. One of the best examples is the ability of 3D system to really match heterogeneity in clinical disease. Here is the example of breast cell lines that are grown in 3D, and all of them really behave differently based on the 3D microenvironment. And you can see from the left-hand side where we have a really defined and organized tumor, which is easy to cure through surgery, as an example. And up to the right, you have a really disseminated tumor that try to invade and produce metastasis. And this is only related to 3D growth, nothing else. However, there is a lot of benefit from 3D development, but in parallel of that, there is also a lot of issue in manipulating, understanding the opportunity given by a 3D model. And the most difficult things is related to having proper readouts, and most of the time, in order to gain much of this 3D assay, imaging is preferred option. However, imaging is containing a lot of information, and then in addition to that, you need to bring on top of imaging data analysis capability in order to really understand what are the real benefits of the 3D technology and what are the mechanism of action or your compound acting in complex models. So I will now move to a few examples, and the first one I want to detail is how we develop a platform to support compound that is supposed to act as an immunogenic cell death inducer. Immunological cell death cycle is really interaction between immune and tumor cells, and at the really start of the cycle you have dying tumor cells, and then they are releasing signal that will bring DC, so dendritic cell, close to the tumor. The signaling from the tumor should normally turn on the DC, and then the third stage of this process is activation of T cells. And normally, if DCs are well activated, they will connect to T cells, activate them, and help them to be specific to the tumor, and then they can go back to the system and fight against tumor cells. What we have developed is multiple readout system where we start quite simple with understanding how cancer cells are dying. And what we want to be sure is that tumor cells are releasing immunological signals. Among them, we know that we can measure secreted ATP, release of markers such as HLG-B1, or change in cellular surface, such as occurring of calreticulin at the cell surface. So that the first steps where we are testing compound, and then we move to a more complex assay where we have co-culture with allogenic DCs and tumor cells. So we have then the ability to measure how DCs are activating by dying tumor cells. And we have then multiple readout on DCs per se, meaning phagocytosis activity, and then change in cell surface marker and cytokine secretion. Then the end of the cycle is how DC is able now to turn on T cells, and then it's another co-culture assay where we have DCs and T cells either coming from mouse or from human, and here what we measure is T cells activation through change in surface marker, change in cytokine secretion. And I want to show you some example from that. So here is the ability of the platform to identify compound that is really immunogenic cell death inducer. So this compound, for the example, is coming from our own chemistry platform. And you can see here that on multiple cell lines, so 2 mouse, 1 and 1 human, that this compound is able to generate secreted ATP. What we want to have is the ability of compound to induce ATP secretion without inducing the full death of the tumor cells. Then from the 2,000 compound, we select 100 having one of these features, and then we move to other readouts, still in the death part of the platform. And we can see that one of the selected compound is able to induce HLG-B1 release from the 3 cell line of interest. And finally, the next step for the cell death part is to monitor cell surface calreticulin expression. And we still have positive signal from this particular compound among 24 compounds screened for this step. And finally, what we've done is to test if this particular compound is able to increase phagocytosis of dying tumor cells by DC, and that's the result we have on the screen here, showing that we have DC positive form CD26 phagocytosis, and we have specific signaling from this DC. Another example where we have closed the cycle is using one of the well-known ICD inducer, dinaciclib compound, where again we work through the platform, confirming that dinaciclib has the ability to induce secretion of ATP, secretion of HEGB1, and calreticulin expression of the cell surface. Then we have co-culture assay again, where we can see that we have increase in phagocytosis capability by DCs induced by dying CD26, and then release of specific cytokine from DC, and then the co-culture where we have dying tumor cells, DCs, and T cells. And what we can see is that we have a polarization of these cells that shift from prominent CD4 population with a starting proliferation of CD8s, and these CD8 are able to start signaling with interferon gamma. So through the platform here, we are able to select compounds that really have this immunogenic cell death inducer profile. With this platform, there are now a lot of options where we can then move with human tumor, with a lot of mouse system in order to also move to in vivo situation with selected compound. I will now discuss a bit on the 3D system. 3D spheroid is really a model that is used for a long time, and they are not now able to be used as a screening system since we are able to have one spheroid per well. This can be even be performed on 384 well plate system, and with really robust assay, having a Z prime score of about 0.6 to 0.7, which is really, really good. Using and playing with 3D system, you can for sure have image-based readout in order to understand how your compound is acting on this 3D structure. And I have here 3 different examples showing that compound could change the size of the spheroid without changing too much the density of tumor cells within the spheroid. In this HEC70 breast model, we can see here that the compound is really destroying the 3D structure, and with really high dose, we cannot evidence anymore organized structure at the end. And in a third example, what we can see is here is that under compound pressure, this MDA-MB231 spheroid is starting to be invasive, and clearly showing that the really well-defined structure as start is really disaggregating and showing that multiple tumors are moving out the spheroid and invade the matrix. It's really good to see this phenotype. However, through image, it's not so easy to measure precise change. Even now, the imaging tools are more and more sophisticated, but readouts on metabolism or viability is still one good system to have improve throughput. And here is the example of using ATP quantification through luminescence, showing that we can have really nice dose response from this 3D system. As an example of middle throughput screening, we have here the screening of 2,500 compounds in two different tumor models, showing that we can identify compounds that show really nice efficacy in controlling the tumor growth and inducing death of these tumor cells in 3D. The improved model for 3D is to have co-culture of tumor cells and immune cells. This is example of tumor cells that are expressing a red marker, fluorescent marker here. So we can see that we have growth over the time of these spheroids. On the left-hand side, there is only tumor cells, and in green, in the middle, you have tumor cells with PBMCs, and on the right-hand side, you have tumor cells in red and T cells, purified T cells in green. Without any activation, you can see that there is no much activity of immune cells against the tumor, meaning there is sure interaction, as we can see that the tumor cells are aggregating around the tumor spheroids. However, there is no killing activity. If you do activate immune cells, we really start to see that immune cells are able to kill the tumor and destroy the tumor quite totally. In the middle, that's still PBMCs, but activated PBMCs, and you can see here that the remaining viable tumor is really low, small viable cells as compared to what we should have in the control here. And with activated T cells, they are able to totally destroy and kill tumor cells. And we can see it through normal light or through fluorescence imaging. So that's the benefits of co-culture system and 3D, where we can really now move to understand interaction between tumor and immune cells. And that's the quantification of that, where we can here evidence that in red, that's the area from the tumor only. In gray, that's tumor plus unstimulated immune cells, and in green, that's tumor plus activated immune cells, where we can really see that there is an efficacy of the immune cells to kill the tumor. And the last example I want to share with you today is what I will call 4D system. And 4D is 3D plus real-time analysis. And we've done some essays aiming at validated various targets. And again, we have done that in multiple steps. The first is selecting the best candidates and the best candidates model. So validating the target expression through mRNA and protein expression, then move to validating our interference system, showing that we did have decrease in mRNA signal when we have the interference in place as compared to the control. Then we have the quantification of that. And then what we've done, having these two tools validated, is to follow the cell growth over the time and confirm that change in cell growth, cell proliferation capability is related to interference and protein expression. And you have here on the right-hand side real-time growth curve showing in red the negative control, in blue the positive control, and in pink, light blue, and green all the target that produce and have an impact on proliferation. And that's on the protein level, showing that the proliferation impact we have is related to change in the target that we are focusing on, meaning we have here the interference that reduce the target and negative and positive control. And really the having the ability to track that over the time is really confirming that you can move that target forward to developing compound against this target. And I think now is the time to Sonia to ask for the poll questions.
Yes, thank you very much, Jean-François. I will launch the poll question right now. It's a simple poll question. We'll give you about 40 seconds to complete. This is done in real time, so your participation is strongly encouraged and very much appreciated. And the question we have for you is, do you currently need cellular assay support in your R&D program? So there's yes, soon, or no. So please go ahead and click on the below, whichever one represents you. And then once we've got the majority of our audience, we will close the polls, share the results, and then we'll pass it over to Olivier. Okay, that looks like it's the majority of our audience. So thank you very much. I'll give a few more seconds for those of you who have not cast your vote. Okay, that looks like it's the majority of everyone. Thank you very much, everyone. I'm now going to close the polls and let's take a look at the results. Okay, so here are the results. We got 24% of our audience is saying yes, 14% are saying soon, and 62% are saying no. So thank you, everyone, for participating in that final poll question. Now I'm going to pass over the mic and the controls over to Olivier. So Olivier, when you're ready, you may begin.
Okay, so thank you, Sonia, and thank you, Jean-François. So I will take advantage of this webinar session to speak about how we can help the drug development with the in vivo models and in vivo pharmacology. So, of course, all the in vivo steps are performed for some parts in parallel of the in vitro. So all is integrated, and we always discuss in an integrated manner all the steps between in vitro and in vivo. It's a complete integrating system. So I would first present our pharmacology platform, and just very briefly. So yes, it's working. So in fact, we have 4 in vivo facilities where we can perform in vivo experiment. So with many different biosecurity level from BSL1 to BSL3, including the possibility to manipulate non-human primate. And of course, all of our in vivo facilities are ALAC accredited and accredited for human sample manipulation, of course, and transgenic animals manipulation also. So during the last 25 years, so we acquired a lot of experience and manipulate many different compounds, probably more than 4,000 compounds that we have manipulate in our labs and develop, use hundreds in vivo models covering probably more than 40 major cancer pathologies. So including all types of tumor models that we have investigated and developed in mice or in rats. Of course, in genic, xenogenic, transgenic, and chemo-induced models with many different tumor growth sites, ectopic, orthotopic, or with metastatic growth. And so all these models are highly characterized and help us to answer many different questions. And we are still developing models and adapt our models to the right question and to make easier to the compound to progress in the different steps. So for sure we are using models for target validation and mechanism of action, characterization, proof of concept, proof of efficacy, and biomarker identification and validation using many different readouts that we put around our models to better understand and qualify the compound that we inject in the models in terms of pharmacology. So classically, we measure the tumor volume, the body weight, the survival, and the metastasis when we have to use metastatic models. We are using large multimodal in vivo imaging, notably to measure the tumor growth development where the tumor are growing in an orthotopic site. We, of course, have developed during the last 10 years many capabilities for genomic analysis. So the NGS, whole exome sequencing, RNA-seq analysis, and including gut microbiota meta-sequencing for some field of applications. So basic biomarkers that we can monitor in liquid biopsy, serum biomarker for sure to monitor the tumor growth in the body of the animals, and also especially during the last year, more recently, we developed the digital PCR that is really helpful to monitor the circulating tumor cells. So in a liquid biopsy. So this is a really nice technology. And finally, so we also have, of course, the possibility to monitor the protein expression in tumor samples by immunohistochemistry for target expression or down-regulation of some proteins for pharmacodynamic analysis, etc. And so complex and multiplex analysis, especially for inflammasome and cytokine analysis using the Luminex system, and of course, as you probably have in mind, many FACS analyses to monitor the immune cell infiltration in the field of immuno-oncology. So this is the short introduction of what is our in vivo platform. And during the in vitro steps, as Jean-François presented before, so we are performing admit-tox studies with so the classical absorption assay with in vivo intestinal absorption, the biodistribution of the compound, with using also label compounds if necessary. We investigate the metabolism of the compound, and just to speak about really nice, interesting technique, the MS2-plex, that help us to monitor the in vivo enzyme-mediated metabolism and transporter, and of course the excretion analysis, which is done classically in this step of drug development. And we are also able to perform toxicities at a later stage. So, and as I mentioned just before, we are using we are able to use radioisotope. So with classical radio labeling with tritium or C14 if necessary, but also with many other isotopes that we can use to radio label, especially antibodies or peptides, to manage the pharmacokinetic and the biodistribution of this type of compound directly using in a non-invasive in vivo imaging systems, with which we can quantify the biodistribution and the pharmacokinetic of such type of compounds using this technique. This is a specialty, I would say, of Oncodesign that has been developed more than 20 years ago. So, and when we have all these results, so we can go through the proof of efficacy of the compound, and depending on what we are knowing on the compound, what is the mechanism of action of the compound. So we will select, I would say, a basic in vivo models just to demonstrate that our hypothesis of mechanism of action are right. And so here are just two examples of basic in vivo models. One is a syngeneic breast tumor models that was used to evaluate the efficacy, the first efficacy of a compound that we expect to impact the immune system of mice to obtain a tumor efficacy. So, and here you can see that the compound didn't show an efficacy on the primary subacute tumor, but you can see here that the compound have a great efficacy on the metastasis, on the lung metastasis that appeared in these tumor models. So it's an interesting results. So on the right side, so you have another example of a classic xenograft breast, human breast xenograft models that we used to compare the efficacy of a new compound, namely here the compound 1, in comparison to a standard of care, which had the same mechanism of action of the compound 1. And you can see here that the compound 1 have a huge efficacy and cured all the tumor in all the animals, and is with a higher efficacy as compared to the standard of care. So, and then, so with based on these results, so we investigate the efficacy of the compound in other models that are normally more representative of the human situation. These models are patient-derived xenografts, and we selected tumors and models from a tissue microarray based on the target expression with immunohistochemistry and select one tumor. It's a triple negative PDX model with very high expression of the target, and you can see that the compound is very active, and then another tumor, a pancreatic tumor with no expression of the target, in which the compound is not effective. So, and here, so based on this data, we showed that tumors with at least 50% of target cells could respond to a treatment. And with this type of models, so it's easy to define, I would say, a cutoff of the target expression that we need to have in the tumor to obtain a significant efficacy of the compound. So, and after that, so based when we have different proof of efficacy of the compound, the question is how to combine the compound with the standard of care to expect entering into the clinic. And so here we have tested, so it's an example with another compound, but we perform the pharmacokinetic profile of the compound after different administration of the compound. That is really helpful to define the treatment schedule and the treatment dosing regimen of the compound that we combine here with the docetaxel, a standard of care that is used in triple negative breast cancer. And here we use human breast xenograft models in the nude rats. And you can see that the combination of the test substance with the docetaxel clearly increased the anti-tumor efficacy and with complete responses. So it's a very interesting results. But during the first studies that we performed with this compound, we observed some suspicious side adverse effects. So in fact, and especially when we administered the compound with IV bolus, repeated IV bolus. And here we try to modify the treatment doses and the treatment administration of the compound by using short IV infusion instead of IV bolus. And clearly we demonstrated that IV infusion of the drug displayed a marked anti-tumor activity and reduced the drug adverse effects. So it's really interesting because adverse effects, of course, it's a major step that we have to avoid for sure. And then, based on all these results, so now let's go to the translational studies to try to better enter into the clinic and to better position the compound into the clinic. So first of all, in this field of work, I want to speak just very briefly about a concept using only one mouse per experimental condition, what we call single mouse preclinical trial. So here, just to show you the feasibility of this concept. So here we have tested two drugs, drug 1 and drug 2, in 12 PDX breast tumor models. And we have 7 to 10 mice per group, classical study, but we compare, so we analyze the individual response in comparison to the majority of the group response. So, and when you pulled the complete responder, the partial responder, and the stable disease in one single responder category, in fact, we obtain 100% of the individual response that match with the majority response of the group. So this is just to exemplify and to try to convince you that this type of preclinical trial is completely feasible, and it's completely feasible to use only one mouse and to test several models in parallel in the same study and to screen the efficacy of the drug in many models in parallel. And here you have an example. It's not in breast cancer, but in colon cancer, PDX models where we have tested monotherapy and the combo FOLFOX, and you can see that the FOLFOX combination increase the number of models that are sensitive to the combo as compared to the monotherapy. And it's clearly the same, so comparable with the clinical situation where FOLFOX increase the outcome of the patients. So, and also based on the on all the genomic data that we require also on our PDX models. So we can use artificial intelligence, data mining, to try to define gene signature of the tumor that should be the most responsive to the treatment, and to better design and select the patients that could enter into further clinical trials. So, and then after, so if you remember, so the drug one that I showed you just 2 slides before, so was very effective in 12 triple negative breast PDX models. And this, the question with this compound was, can I treat other types of breast tumor patients with my compound? So, and the question here, can I treat luminal B breast cancer? So, and how can I combine my compound with the standard of care that are classically used to treat this type of patients? So mainly the palbociclib and the fulvestrant. So, and here you can see that the drug one clearly synergizes with the palbociclib or with the combination of palbociclib plus fulvestrant. So it's a very nice, interesting results, because it helps to extend the possibility of positioning the compound into the clinic. So, but then after the other question was, what about the patient that become resistant to the hormone therapy, which is the majority, unfortunately, the case in the patient that are treated with hormone therapy for many years. So, and to answer this question, so we developed a model that acquires resistance to fulvestrant treatment. And here you can see that this model is completely resistant and is able to grow in mice without estrogen and with fulvestrant, sorry. And we didn't see any effects of the fulvestrant, whatever the dosing regimen we used. We didn't see any effect of the fulvestrant arrest, and no effect also of addition of estradiol, meaning that we obtain a completely resistant luminal B PDX model that is completely resistant to fulvestrant treatment. And so this model was, of course, completely characterized with genomic analysis, and I will not describe in detail this slide, but just to show you that we identify a cluster, the cluster 1, that is completely correlated with the tumor that are treated with fulvestrant. So, and we identify with RNA expression analysis that the pathway that are upregulated clearly correspond to the pathway that are known to be correlated with the hormonal resistance of the tumor. So, based on these interesting results, so we use this. We are now using this resistant model to try to position the drug one in this type of resistant tumor in the clinic, and then treat the tumors with the drug one and try to see if we can restore the sensitivity of the tumor to fulvestrant after the treatment of the drug one. So unfortunately, I don't have the data in my hand today, so it's still ongoing. But just to show the concept of the strategy that we have tested. So, and during this presentation, so I speak a lot about syngenic and xenogenic models, but what about the evaluation of compounds that are targeting the immune system and that are not cross-reacting with the immune target? So, in fact, in this case, we have developed lots of immune-humanized mouse models, and we have plenty of models. But here, just to show you the interest of these models when we combine immune-humanized mouse models with PDX models. And here we have investigated the immune infiltration of the tumor in a panel of breast and ovary patient tumors, from which we succeed in the establishment of PDX models that we have engrafted in humanized animals. And we have clearly shown that the tumor that are poorly infiltrated in the humans remains poorly infiltrated when the tumor are engrafted in immune-humanized mice. And inversely, the tumors that are highly infiltrated in patients are also highly infiltrated when engrafted in humanized mice. So, but unfortunately, when we tested the pembrolizumab on the higher infiltrated PDX tumors, we didn't succeed in showing the anti-tumor efficacy. But based on the FACS analysis of the immune infiltration, we are able to monitor a modification of the tumor infiltrating lymphocyte after the treatment with pembrolizumab. So meaning that the compound which the cells and the target and perform the job, but it's not translate into tumor size reduction. So, and very briefly, so today about immune humanized animals, we are working with a heterologous system. We are using PBMC or immune cells that are coming from a donor and the tumor from another donor. So it's really helpful, but now we are trying to develop more predictive models with autologous system where immune cells and tumor cells are coming from the same patients, and in parallel we developed PDX from with the tumor, and develop IPS with PBMC coming from the same patient, that we are using to obtain hematopoietic stem cells that we inject in mice to reconstitute an immune system coming from the same patient of the tumor. So just to mention here that we are able, and we are always trying to innovate and to adapt our models to the specific compounds that you want to develop. And depending on all the data that you acquire internally or that we acquire all together, for doing that, of course, we have to anticipate and to work in a very close relationship to establish altogether the strategy with your team. So we are dedicating a complete team that is working with your research and development team to optimize all the strategy and the timing and of course the budget. And for sure, our main goal, as all of you, is to try to reduce the timing to get very nice and very efficient compounds, and that will be very helpful for the patients that are waiting for that. So thank you very much. It's the end of my presentation. So thank you all for attending the meeting.
Well, thank you very much, Olivier and Jean-François, for that very insightful and detailed presentation. I hope everyone enjoyed it. Now we have time for our last poll question and maybe one question from our audience members. So let me launch our last poll question. And there it is. So remember, this is done in real time, so your participation is strongly encouraged and very much appreciated. And it's a very simple question. What part of the drug discovery process do you most need support to speed up your discovery? So you have two options below: specific studies at any stage of the drug discovery journey, or a comprehensive support from in vitro assays to the in vivo models. Okay, and I'll give you a few more seconds to complete that that poll. Looks like that's the majority of everyone, so thank you. I'm going to close it now, and let's take a look at the results. Okay, so the results are here. We have 33% of our audience are saying specific studies at any stage of the drug discovery journey, and then that's 33% of our audience. And then we have 67% of our audience saying comprehensive support from the in vitro assays to the Vivo model. So thank you everyone for participating in our final poll question. And now we're going to start with the Q&A. We have time for maybe one question. So I'd invite Olivier and Jean-François to put on their webcams. There I am, everyone. So remember, you can send in your questions by using that questions window located on the right-hand side of your screen, and we'll address your questions after this presentation. So let's start with our first question here. This audience member is asking, you did not mention any organoids capabilities in the presentation. Is it something validated at Oncodesign? Who would like to take that one?
Yeah, so I will take that one.
Okay.
So yes, we do have organoid capability in-house. However, there is nothing I can share today with you, so that's why there is nothing in my presentation. What I can share with you today is we've worked with brain-derived organoids. We did not establish them, but we have produce them and use them in various assays. And we have two programs, one in partnership with an academic collaborator trying to establish and engineer pediatric tumor-derived organoids. And the other one is an internal program to develop our own organoids and have them ready for testing your compound or our compound that is developed internally.
Mm-hmm. Okay, thank you very much, Jean-François. I'm going to squeeze one more in here. Are you able to run in vitro studies with primary cells? This audience member is asking. That'll be our last question.
Oh, yes, for sure. So we are Bio Research Center accredited in France, so we can receive samples from healthy or diseased donor. And we have established a lot of partnership with hospitals in France or in Europe to collect patients in an ethically authorized manner and have them use in vitro or to test target expression or perform mechanism of action related studies.
Okay, well, thank you very much, Jean-François, for answering that. Well, thank you very much for those questions. We have reached the end of the Q&A portion of this webinar. If we couldn't attend to your questions, and there's a number of them there, the team at Oncodesign will follow up with you after this presentation. And if you have any further questions, please reach out to them at contact@oncodesign.com. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from X-Talks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is strongly encouraged and very much appreciated, as it will help us to improve on our further webinars. Now, give me a few seconds. I'm going to send you a link in your chat box, and you'll be able to view the recording of this event and also share it with your colleagues once they register for the recording as well. So I encourage you to do that and get even more out of today's presentation by downloading the supporting material that is available under the Handouts tab, and it's a PDF of today's presentation. So please go and check that out. Now, please join us in thanking our speakers, Jean-François and Olivier, for that very insightful presentation. So thank you very much, gentlemen.
Thank you, Sonia, for your support, and thank you all for attending this webinar.
Thank you. On behalf of the team here at X-Talks, we hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at X-Talks, again, we'd like to thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now. Thank you very much, everyone. Thank you, Jean-François. Thank you, Olivier. All right, bye-bye. Bye, everyone.